Okay, Mendel genetics ratios are crystal clear. We can get monohybrid, dihybrid like this. When it comes to incomplete dominance, when it comes to codominance, 1-to-1 ratios we can expect. Now, when it comes to the other concept, linked gene, what is the ratio? We can't get a very clear patterns of ratios for certain phenomenon like crossing over. The other phenomenon is coming epistasis. So when it comes epistasis, there again there is a pattern of ratios. But luckily, these pattern of ratios we can identify under epistasis. So then we can clearly put into the concept, okay, question is based on epistasis. And then we can try to find the solution. But at last, we never find a pattern of ratios. Then we have to make a conclusion. This is linked gene. The ratios are coming because of the linked gene. So I think I can get some questions, such questions.
Sir,
Yeah.
so in the second situation, if crossing over take place, won't they simply go to the first one and capitally come to the right one?
This is actually this chromosome separate to one gamete, and this chromosome separate to one gamete. Because here only we have two chromosomes, not four. Here we have four chromosomes. Here we have two chromosomes. Because when we have four chromosomes or two homologous pair, then we try to get the combination. But we don't have a chance here to get the combination. It is clear this chromosome is going to one gamete. This one is going to one gamete. And here we can make the combination because this chromosome in the first situation, this chromosome and this chromosome can join together. So this one and this one can join together. So we can find some combinations. We have freedom to get the combination, but we don't have a combination here. Just only one, and it's going to the other one. So, and if A and B on the same chromosome, they go to the one gamete and no separations. So in the third situation, we make another concept, can happen still due to crossing over. But if they are very close to each other, then there's no chance of separation. So in linked gene, we define it, alleles are on the same chromosome and they are close to each other. And they are not, they are inherit together. Alleles are on the same chromosome, they are close to each other, inherit together. So these 3 points we can raise under linked gene. All right, so if you look all the 20 years of questions, they haven't made the question to cover up each and every concept. So questions are made from prominent concept. You will see when you are doing all these questions in the past papers. But we understand all the concept and we try to prepare. Any questions come, we have a plan, we have a algorithm to find the answer. So it's like a filtering process, whether it's Mendel or non-Mendel. If it is non-Mendel, what is the phenotype? What is the ratios? So from this phenotype and ratios, can I identify the concept? If I identify the concept, then we try to solve the problem. So this is the algorithm: Mendel or non-Mendel. If it is non-Mendel, what are the ratios and phenotype, and what could be the concept? Like that, we try to move step by step to solve the problems. This is another concept which is also not fitting to Mendel. So we consider as an non-Mendel condition human sex determination and inheritance of X-linked gene. Right. So we understand it like this. If you take a human cell, there are 23 pairs of chromosomes. And these 23 pairs we divide into 22 autosome and one sex chromosome. So autosomes responsible for all kind of characteristics of human, and sex chromosomes responsible for determination of gender. That is male or female. So at the moment, I only take male and female. If there are the other combinations come up, we just forget about it. At the moment. They are not important at the moment for the learning purpose. So male and females. So when it comes to male and female, we can understand the chromosomes, they have genes. So if this is a chromosome, this chromosome has many genes. These genes make proteins. These proteins are responsible to decide the male. Because male, the phenotype are different, and there are many different proteins and different structures are there. And the female one, same, there should be a chromosome for female one. The female chromosome should also decide the gender by expressing proteins, set of protein expressed during the development of fetus, and then from the fetal development, it make the male or female. So when you look at these male and female chromosomes, this represent a female one, and this represent male one. And this is number-wise 23. The sex chromosome has number 23. And male one has a name called X, female has a name called Y. Male has X chromosomes, Y chromosome, female has X chromosomes. And if we look at this homologous chromosome, we can also find another different. So we call homologous chromosomes, they are very similar in shape, and they have more similarities, because if one chromosome has the 1,000 gene, the homologous one on the other side, they have 1,000 genes. But here is a different. X1 is bigger, Y1 is smaller. They are not completely same in the shapes and not complete in the gene number. So X is bigger, it can have more genes. Y is smaller, it has only small amount of gene. This explains females are complicated because they have more genes to express. And Y, less. On the top of that, X chromosome is bigger. It has more space for other type of genes. Not only sex determination gene, but different other characters can also present in X chromosome. So in this case, how we can define the sex? So female is always X and X, and male is X and Y. Now this explains why in human population or maybe other animal populations, male and female are 50-50. So if we draw Punnett square, X, 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 Y, X, 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 Y, X, Y. So 50% chance of getting a female, 50% chance of getting a male. That means a father and mother, XX mother, XY father, when they want to have a children, the chances are become male and female is half. So this is the genetic background of a sex determination in human. Now we have a additional step from this one. Additional step is if this X chromosome has still more space for other genes, how these genes are responsible for the inheritance. So this part is not responsible for sex determination. There are small additional bonus space. This bonus space is provided to add other type of genes. As a result, we have to discuss sex determination and inheritance of X-linked gene. So let's see what is X-linked gene. Now we take the X chromosome, genes which are not responsible for sex determination is called X-linked gene. X chromosome linked gene are additional genes responsible for different type of characters. Now, why it is important to discuss this X-linked gene? There are common disorders in human population are coming from X-linked gene. So let's take one example of this. Just 1 second. Yes. Because I need more space to write. Yeah. Right, we have to discuss a common condition. This condition is called red-green color blindness in human. Now, this condition is called red-green color blindness. What is the symptoms of conditions? It is difficult to distinguish red and green. When the red and green are together, it's very difficult to distinguish red color and green color. So the people have difficulties to clearly identify the picture and clearly identify the color when they have these conditions. So red-green color blindness is X-linked. Now this is due to a mutation of the gene. And it is a recessive. That means capital B no color blindness. Simple B color blindness. Red and green blindness. And this is red and green blindness. Right? No red and green blindness, capital B. Simple B, there is a red and green color blindness. So we try to make this situation like this. Mother, X and X, and father, X and Y. Now these genes are only on X chromosomes, not Y chromosomes. Father is healthy. Father is healthy. It has capital B. Mother is also healthy. Capital B. But mother has a recessive one as well. Now parents are healthy. When parents are healthy, what will happen in children? Capital X, B, X simple B, X capital B, and Y. Now genotypes are X capital B, X capital B, X capital B, X simple B, X capital B, Y. X simple B, Y. Now there is a problem. That means there's a chance. What is the chance? Chance is 1/4. The probability is 25%. 25% of chance a child get color blindness. There is another interesting thing we can notice from this Punnett square. Can you guess what it is?
The females are healthy and the males, there's a chance, 50-50 chance.
Exactly, yes. Now, this kind of X-linked gene, males are more prone to get the disease. Males are more affected because males only one allele enough to decide, right? Only one allele enough to decide phenotype. That's one reason maybe males are very bad in shopping. if they have most of them are colorblind, they can't select good colors. Yes, it is true. If we look population, human population, I think uh, 2% or 2% or 3-4% males are colorblind, and like 0% only females are affected. Very less females affected, but out of 100, maybe a few people are affected with color blindness in the population, because males are more affected with this color blindness. It's not a big bad conditions, just only the problem with identification of colors, and mostly this goes unnoticed until the man is going for a specific interview or trying to identify some kind of red green color blindness, and he complain about this. Then we can notice why is that, because I had a one student accidentally, if I write on the whiteboard with red color, and he complains, I can't see well this red color. So it's very difficult to see the red color like that. He complained like that. Finally, there's a small test online. You can have some diagrams. From these diagrams, we can decide whether person is colorblind or not by reading the numbers of these red and blue charts, red and green charts. Another conditions common, and that is not good as red green color blindness. This condition is called hemophilia. Now, hemophilia is a condition in which the person is unable to clot blood. So, defect in blood clotting. So, have you learned blood clotting in the circulation lesson?
Yes.
So, if there is a small injury or small damage to blood vessel, then there is automatic mechanism within a short period of time. It make a clot and then prevent bleeding. So, but most of the time, when we learning this blood clotting system, we don't learn the complete blood clotting pathway. We learn only on our syllabus, A-level syllabus, usually for all kind of a syllabus. We learn only the part of the blood clotting, but when you enroll to the like medical degrees like things, then complete blood clotting pathway you are learning. It's a very complicated process. So it has kind of a small dominoes like effect, like A goes to B, B goes to C, C goes to D, D goes to E, E go to F, like that. One after another, the event happens. So mostly we are learning in our systems the last few events. So what we learn actually, the last few events are prothrombin, thrombin convert fibrinogen to fibrin, make a mesh, and fibrin can make a clot. So starting mostly with the prothrombin level, prothrombin to thrombin. There are blood proteins. These blood proteins activate, and then activated blood protein activate another protein until it make a insoluble proteins. But before it happen, there are many other factors are involved. The factors are proteins. So in hemophilia, there is a mutation in one gene. This mutation cannot produce factor 8. The factor 8 is required for blood clotting. There is a mutation on the gene, cannot make that proteins, and they have a defect in blood clotting. So this is again X-linked. Situation is very similar. If father is healthy and mother is having this defective alleles, again 25% of chance the male child get the disease.
So, in this hemophilia, if they bleed, they won't stop bleeding, sir.
You mean continuously bleeding?
If they bleed, they won't stop it like continuous. Yeah.
I didn't hear you properly. Can you repeat?
Yes, a continuous bleeding, sir. Do they undergo continuous bleeding?
Uh, well, it is like this. Now, in a healthy condition, like us, we never feel any kind of a bleeding unless there is a wound, injury, or damage. So we have experience always if we cut the finger and maybe uh, if we have a small injury, then there's a bleeding. But you know, certain cases, maybe we experience, some people has nose bleeding. There are a couple of cases we experience, sometimes friends and some other people, they have a nose bleeding. If person is healthy, it doesn't mean the blood vessels are damaged, because there is a chance always our blood vessels are damaging, like capillaries, they can damage. Because when we eat something hard stuff, there is a damage in the mouth, but we don't notice it. Probably when we hit a little bit harder our body to certain surface, there is a small bleeding under the skin, but we never notice this. Between joints, when joints are moving faster, there could be chance of capillary damage, but we never notice these things because automatically these minor bleeding are prevented by the blood clotting system. That means continuously there is a chance of having a minor bleeding in healthy conditions, but this is not happening. It's automatically controlled by the system. Now, people with hemophilia, it's not the case. There are minor damage happening when they eat, rubbing the mouth with the food and other things, the body and then the joints. There is a minor bleeding, but they can't control this minor bleeding. So they have a excessive bleeding, like small bleedings. It doesn't mean it's 100%. It's not clotting. So prolonged clotting, they tend to lose the blood from the joints, mouth, and other under the skin, which is not the good things. It is painful condition. On top of that, if they make an injury, if they lost tooth, because tooth, everybody is losing their tooth, the kids and maybe after elder time, then there's again bleeding. Even kids, we have experience when we lose our teeth, there is a bleeding. Small bleeding, we feel the taste of the blood, everything is coming to the mouth. And hemophilia people, they have to suffer a lot because they have an excessive bleeding and they have a very risky if there's an injury, then it is very difficult to stop the bleeding. So that is the problem with hemophilia. Yeah, so we can notice these people nose bleeding, skin bleeding, and continuously they suffer some kind of a discomfort of the bleeding. Finally, they have to treat for these conditions. If they have a problem of bleeding, they have to continuously take some kind of drugs which prevent hemophilia. So hemophilia is again high in male, and it is again disorder with X-linked, and you can also notice some of the things like mother is the carrier and father is healthy. Some of the common disorders like things should be screened, should be checked before the marriage. That's another story. How we can detect the carriers, how we can detect defective genes come under different levels. All right, so we have a last two concept of non-Mendelian genetics. So if you recall what we have learned, incomplete dominance, codominance, multiple alleles, and linked gene, sex, X-linked gene, sex determination. These are the things come under non-Mendel. The last two things to finish non-Mendel genetics. I can say the biggest part of the genetic is Mendel and non-Mendel genetics. After that, others are again normal concept and theories. So this is the most important part. And if you look questions asked from last 20 years, based on Mendel and non-Mendel genetic, 80, more than 80-90% come from this place. Others are more just like a concept. So it's easy to answer those questions, but hard part coming from this. Right. We continue the last two concept. Before epistasis, we have this condition called a pleiotropy. Now, pleiotropy, how we can understand this? We'll take this as example. Hemophilia. What is the problem? There is a gene. This gene is mutated and cannot make the factor 8. They have problem in bleeding. But pleiotropy, I can explain like this. There is a gene, there's a mutation. After the mutation, the protein is defective. As protein defective, the person person show many different kind of symptoms and signs all of the body. So bleeding is one, just bleeding, but a person is showing many different problems: digestive system, reproductive system, respiratory systems, heart, kidney, that the whole body is involved, everything is affected. One gene, but many things are affected of the body. So this is the pleiotropy. So this X is a one gene. As a result, this character trait is affected. This one is also affected, and this one is on. So pleiotropy explain defect in one gene cause problem in many parts of the body. But here we can't say one gene, the problem is bleeding. But at least bleeding, it can happen most of the part of the body, but it's bleeding. No second thing is not coming, bleeding. But when it comes to the pleiotropy, the whole part of the body involved, maybe he can develop kidney failure, he can develop diabetes, he can develop infectious disease, he can develop respiratory problems, infertile, like so many things affected. So on the pleiotropy, we are going to learn how can effect of one gene make problem all over the body. Okay, there are two conditions we have to learn as per our resource book. The first one is called sickle cell anemia. Right. Anemia is lack of blood, less amount of blood. Sickle cell mean the red blood cell has sickle shape. Otherwise, healthy red blood cell has this biconcave shape like this, but sickle cell is like this, very elongated shape. Why red blood cell get this shape? If the red blood cell get this shape, why many part of the body is affected? We're going to learn under this sickle cell anemia. So it's a defect in one gene, just only one gene. But effect is many part of the body and many different signs and symptoms. Okay, we are coming to the end of the non-Mendel genetic slowly, and we'll take next week again 1 or 2 questions from the past papers, because we have done a few things under non-Mendel genetics, and we can keep on doing questions. I think there's another tute I have to send you, still non-Mendel genetics. They are under non-Mendel genetics, but it doesn't matter. When we are doing general topics, we can discuss, right? Okay, I'm going to stop here. We'll continue again next week, right?
Okay.
Okay, see you.
Thank you, sir.
Okay, bye.
Bye.